Hello, I am an AI voice, Alice. Today I will introduce you to the Plant Contact Angle Experimental Guide by Professor Q from the Department of Biotechnology at Korea University. The goal of this experiment is for students to understand how the wax and surfactants on plant surfaces contribute to crop protection. Additionally, it provides an opportunity to directly observe this through an engaging experiment using a mobile phone. First, what is plant surface wax? Plants have a layer called the cuticle on their surfaces. Depending on the plant, the thickness of the cuticle layer can vary, typically between 200 nanometers and 50 micrometers. On the exterior of this cuticle layer is a thin wax layer known as epicuticular wax. This wax can be coated like a film and sometimes forms three-dimensional crystalline structures. The wax layer performs very important physiological functions. Epicuticular wax primarily prevents water loss and acts as a physical barrier against external pathogens and pests. It also protects plant cells from ultraviolet rays and contributes to increasing the plant's resistance to specific environmental stresses, such as drought and high temperatures. The composition of plant surface wax is primarily made up of various lipid-like substances, such as alkanes, fatty acids, alcohols, and ketones. The composition and structure of these compounds vary from plant to plant, which in turn determines the hydrophobic characteristics of the plant surface. For example, some plants exhibit a feature where moisture does not stay on the surface but rolls off. This property, known as hydrophobicity, is due to the epicuticular wax. For instance, the white substance on the surface of plants like kale and grapes is not pesticide or sugar, but rather this epicuticular wax. This wax layer provides an essential defense mechanism for plants to survive in external environments. Recent research has shown that this wax layer also prevents the adhesion of pathogens like Escherichia coli and Salmonella that cause food poisoning. However, the hydrophobicity of the wax layer makes it difficult to uniformly apply pesticides to plants. If pesticides do not adhere well to the plant surface, uniform application becomes difficult, and the effectiveness of the pesticide can be reduced. A prime example is the lotus effect. The surface of the lotus leaf is extremely hydrophobic, causing water droplets to easily roll off and keeping the leaf consistently clean. This property is known as wettability, and it is quantitatively measured using the contact angle. Various products that mimic the lotus leaf effect have been developed using this natural phenomenon to create water-repellent items, such as shoes, paints, and even outdoor clothing. By ensuring that water droplets do not adhere to the surface, these products stay dry and clean for longer periods. For instance, shoes treated with lotus effect technology are more resistant to water and dirt, making them ideal for wet environments. Scientists, inspired by the unique self-cleaning properties of the lotus leaf, have applied this concept to fields such as nanotechnology and material science, leading to experiments that aim to create more durable, self-cleaning surfaces for everyday use and industrial applications. The contact angle is the angle formed between the plant surface and a water droplet, measured based on 90 degrees. Surfaces with a contact angle less than 90 degrees are defined as hydrophilic, meaning water adheres well, whereas those with a contact angle greater than 90 degrees are defined as hydrophobic, meaning water does not adhere well. A contact angle greater than 140 degrees, such as on lotus leaves, indicates extreme hydrophobicity. This contact angle is closely related to the plant's surface structure. For example, kale has a folded cuticle layer with three-dimensional wax crystals formed on it, displaying strong hydrophobicity with a contact angle of more than 125 degrees. Conversely, vegetables like lettuce typically have hydrophilic surfaces that adhere well to water. But as they undergo drought stress or enter the flowering stage, the wax layer thickens, increasing the contact angle to around 100 degrees. These surfaces have only three-dimensional wax crystals. Arugula or regular lettuce, on the other hand, do not have wax crystals, but only a film-like layer. In this case, the contact angle is around 50 degrees. Surfactants play a critical role in effectively applying pesticides on these hydrophobic plant surfaces. They help dissolve pesticides that do not mix well with water and ensure that they adhere well to hydrophobic surfaces. Using surfactants can maximize the effectiveness of a smaller amount of pesticide, contributing to environmental protection. Precision experiments to measure contact angles typically require expensive laboratory equipment. However, without using such costly devices, students can perform the experiment simply using their smartphones. Previously, contact angles were measured using software like ImageJ, but now mobile applications such as Angulus or Anglometer 360 allow for fast and convenient measurements. These apps significantly reduce the time required for students to complete the experiment. The materials needed for the experiment include kale or broccoli with a thick wax layer, scissors, double-sided tape, a paintbrush, water, and 0.1% soapy water, acting as a surfactant. The experimental procedure is as follows. First, a droplet of water is placed on the unaltered waxy surface of the broccoli leaf. Then, the wax is removed using a paintbrush, and water is dropped on the cleaned surface again. Lastly, soapy water is applied to the wax-removed area. After each treatment, a photo is taken, and the contact angle is measured using the Angulus app. In the experiment, when a water droplet was placed on the surface with wax, the contact angle was approximately 130 degrees. After removing the wax, the contact angle decreased to below 90 degrees. When soapy water was applied, the contact angle further reduced to about 50 degrees. Through this, students can directly observe the effect of plant surface wax on water wettability and understand how surfactants interact with hydrophobic surfaces. This experiment also provides an opportunity for students to learn the basics of statistical analysis. The results can be analyzed using paired sample t-tests, and a p-value less than 0.05 indicates that the difference between the two groups is statistically significant, meaning the difference in contact angles between surfaces with and without wax is real and not due to random chance. Additionally, the difference in contact angles with and without soapy water on wax surfaces can be scientifically analyzed to assess the effect of surfactants on plant surface wettability. This experiment is designed using the one variable at a time method, where only one variable is changed at a time to observe its effect on the results. This helps control other variables and ensure accurate results. This analysis process aids students in understanding statistical techniques and developing scientific thinking skills. Furthermore, by using smartphones in the experiment, students actively participate in research and collect data independently. Various sensors built into smartphones enable a range of experiments, such as quantifying color changes in plants. For example, students can use the smartphone camera to capture plant images and analyze RGB values to accurately measure changes in plant color and concentration. Finally, after completing the experiment, students fill out a survey to assess their understanding of the experiment and the role of surfactants. Most students reported gaining a better understanding of the role of plant surface wax and surfactants through the experiment and responded positively to the use of mobile apps in the experiment. This educational approach using smartphones provides students with deeper learning experiences and enhances real-world application possibilities. This experiment serves as an effective educational tool that helps students understand the characteristics of plant surfaces and contributes to crop cultivation and environmental protection. Thank you for listening.